Assalamualaikum to my panel, Dr. Izzati and Dr. Amaliawati. My presentation is titled Isolation and Characterization of Drought-Tolerant IAA-Producing Bacteria from Soil from Chuping District. My name is Nur Ali Natasha Mdidizol, student ID 2023-298146 from the Applied Microbiology Program, and this project is supervised by Parafida Abdirasul. I am honored to present my research to you today. As for introduction, climate change is making drought more severe, with 2024 that has been recorded as the hottest year in Chuping Perlis with 40 degrees Celsius. This strengthens agricultural sustainability, especially in tropical countries like Malaysia. My project addresses a specific local problem, the Chuping district in Perlis, an area where water scarcity limits forming. While research on drought-tolerant bacteria is active in India and Africa, there is a significant gap in identifying locally adapted strain right here in Malaysia. Therefore, my research aims to answer two key questions, which is first, how many drought-tolerant IAA-producing bacteria can we find in Chuping soils? And second, what specific species are they? To achieve this, I have three clear objectives, which is to isolate these bacteria, to identify the most promising one, and to characterize their IAA production potential. The significance is straightforward. This study will document the first collection of PGP bacteria from Chuping's unique arid soil. The ultimate goal is to contribute knowledge that could lead to more sustainable agriculture and healthier soil in drought-prone regions. This idea is built on a strong scientific foundation. We know that certain bacteria called plant growth promoting bacteria or PGPB can help plants cope with drought in a remarkable way. They work through two main types of mechanisms, directly by producing hormone like IAA which reshape roots for better water uptake, or by helping plants uptake nutrients. Indirectly, by reducing plant stress level. My project focuses on the direct mechanism of IAA production. Study confirmed that IAA is a key player in modifying root system to enhance drought tolerance, so I am targeting a proven crucial bacterial trait. Despite this global knowledge, a clear gap exists. No study has specifically targeted the isolation of drought-tolerant IAA-producing bacteria from the arid soils of Chuping, Malaysia. This makes our location not just a site, but a unique opportunity. The high-stress environment means there is high potential to find uniquely adapted bacterial candidates. To find this microbe, I will collect soil from Chuping at 0 to 20 cm depth. This soil will be immediately preserved on ice and stored at -20 degrees Celsius to maintain the native microbial community. For the initial isolation, I will apply selective pressure from the very start. Using the zero dilution technique, I will put the sample on tryptic soy agar amended with 18% PAG 6000. This is a critical design choice. By introducing the drought stimulating agent directly into the grow medium, this first step will filter out sensitive microbes. I will then purify this primary isolates through repeated streaking on the same pH containing media to obtain pure culture for the next stage. The second stage will involve precise quantitative assay to rank the isolates. I will quantify drought tolerance in liquid culture. Each isolate will be grown in tryptic soy broth with a gradient of pH concentration, creating osmotic potential from -0.15 MPa to severe -1.2 MPa. Growth will be monitored spectrophotometrically over 48 to 72 hours. Based on the optical density at the toughest condition, which is -1.2 MPa, I will classify each isolate as sensitive, tolerant, or highly tolerant. The top-performing drought-tolerant isolate will then enter the IAA production screen. This isolate will then be cultured in tryptophan-supplemented broth to induce hormone production. I will then use the Sarkowski colorimetric assay. In this test, mixing the bacterial supernatant with the reagent produces a pink color if IAA is present. The intensity of this color measured at 520 nanometer will allow me to quantify the concentration of IAA produced by each strain. Only isolate that grow under severe drought and produce significant level of IAA will then be selected as the promising PGPB candidate. For this promising isolate, they will undergo comprehensive physiological profiling that include documenting colony morphology. To continue, the isolate selected for both drought tolerance and IAA production will undergo a series of characterization to build their full profile. First, we have microscopic characterization. The primary technique here is gram staining. This will allow me to observe the basic cell shape and determine their fundamental gram reaction, which is a critical first step in bacterial classification. Second, which is the biochemical characterization. To gain more specific physiological data, I will perform key biochemical tests, including catalase and oxidase. The result from this test, combined with the microscopic data, will give a preliminary genus level identification. This gives information in the functional group of the isolate belong to. Finally, which is the molecular identification. Genomic DNA from the promising candidate will be extracted, and the 16S ribosomal RNA gene will be amplified via PCR using universal primers. Sequencing this gene and comparing it to the international databases like NCBI BLAST will provide a species-level identification. By combining all the data from the phenotype to the genotype, this will lead to the final identification of our target bacteria, which is drought-tolerant bacteria that are capable of producing IAA. Then, from this entire process, I expect 3 outcomes, which is 1. A pure culture collection of bacteria from Chuping soil. 2. The identification of strain with high drought tolerance, specifically those growing best at the severe negative 1.2 MPa condition. And 3. The quantification of their IAA production. I am also aware of practical challenges, which is contamination risk is always present in microbiology, and bacteria can behave differently in a lab plate as compared to real soil. My mitigation plans are direct, which is by implementing a strict aseptic technique and using multiple types of growth media to reduce cultural bias and capture broader spectrum of native microbial community for a more promising result. Finally, this Gantt chart outlines my project timeline. I am currently at the submission of proposal and presentation. The plan ensures all activities are completed systematically before the final submission. In conclusion, this project bridged a local agricultural challenge in Chuping with a targeted scientific search for microbial solution. By systematically isolating and characterizing drought-tolerant IAA-producing bacteria, it's aimed to lay the groundwork for sustainable, bio-based strategies in our region. Thank you, Dr. Izzati and Dr. Amaliawati, for your time and consideration. This concludes my presentation.